carried away. There we go. So we were talking about how populations change, but now we're going to be moving on how to populations change enough. So now it's not just one population of a species. It actually ends up as two species, right? So, and of course, the first thing we have to do if we're going to decide when two species are two species, this is species A and this is species B, is what differentiates them, right? What makes them different? And I started off by mentioning to you guys that the way they look, right? The very first basic thing that you learn to recognize as a human being. You can tell a tree, from a cat. You can tell a dog from a cow. It's that morphological differences. But then we went down to a little bit more detail, and we incorporated Ernst Mayr's definition. This is where it got a little bit tricky, especially when you look at the part that's in the red of his definition. What they say is if you're a member of another species and you reproduce and you broach all of those barriers that we were looking at last time in class, and you produce viable fertile offspring, right? Or if you do not produce viable fertile offspring, then you are members of different species, right? And those reproductive isolating barriers I talked about, all of these things in between aside, obviously these are organisms that represent the fact that, we are in between the rungs of the ladder, that species don't arise overnight and are completely different. There are many cases, in some of the critters we looked at, in which you got organisms that are kind of in between, like wogs and mules, right? They're hybrids that are occasionally, or most often in the case of the wogs, born fertile, and it even happens in the wild amongst, like I showed you with these critters. All right, so we get over habitat isolation, temporal isolation. Basically, in the first part, they're prezygotic barriers. What we're basically doing is preventing organisms from even getting to the point where their gametes come together, right? At the very end, we have gametic isolation. But prior to that, in the prezygotic barriers, it's like I've said before, the way you're going to keep these organisms separate, or the way you're going to, once they become separate, the way you're going to reinforce that. In other words, the way you're going to eventually drive, I left my pen at home. I can use my finger. The way you're eventually going to drive even further and further distance between these two species. Remember, there's their common ancestor. Remember, we did some exercise yesterday, so the dogs are right about there, horses and dogs and wolves, horses and mule or donkeys a little bit further down, right? But what we're saying is once you get beyond all these barriers you have here, then eventually you reach the point in terms of the prezygotic where fertilization can happen. Assume that the gametes can bind together and the sperm can fertilize the egg, you're going to get a zygote. But it doesn't stop there. Remember, there were three conditions or three hurdles that had to be cleared on that end. For example, the hybrids don't live very long in some cases. So this would be essentially the dead end street for this genetic information, right? So there's habitat, there's when they reproduce, there's if there's a behavioral isolation, a mechanical, right? This is as simple as engineering. If tab A doesn't fit into slot B, then you're not going to get reproduction. And I mentioned the gametes, right? But reduced hybrid viability. So Ranapippians and Rana sylvatica, very closely related to each other, so close that they can make hybrids, right? So they can produce offspring, but they don't produce viable fertile offspring. So by definition, then they'd be two separate species. Or if the hybrids are viable, and like in the case of the mule, remember, many times they're actually very viable. That's what we know about mules. Mules are very strong animals, right? So you get viability, but you get sterility, or you get usually sterility. Again, remember we said that there are those exceptions, and to be honest, those exceptions are important to remember because those exceptions always remind us that separate creation, that individual arising of I'm not using the word right, but you get the idea. Individual arising of species lines is not the way it works. We come back from that universal common ancestor. So we have these critters that fit someplace in between. And finally, I said, all right, so maybe you get viable fertile hybrids, but their offspring are not viable. All right, so you get a little glimpse of the family tree being perpetuated into history, but as soon as those hybrids have offspring, then those offspring don't survive. All right. Now, if an organism is able to clear all those hurdles, if two organisms are able to clear all those hurdles, then by Meyer's definition, they are considered the same species, right? Otherwise, they're two separate species with all the exceptions that we talked about fit in between, right? The horse and the donkey and all that stuff. All right, now patterns of speciation. Now, patterns of speciation, it's easy to get this a little bit confused with those reproductively isolating mechanisms, because especially allopatric, right? Allopatric speciation only is going to involve habitat isolation. In other words, that very first pre-zygotic barrier, that keeping them apart, you know, the species of Spartina grass on either side of the Atlantic Ocean, if you keep these organisms far enough apart, then it's just a physical thing, right? Organisms can't reproduce unless they're in the same general area. There's a very interesting story I'll tell you about how barnacles, an organisms we'll study about later on, how barnacles get around this problem of being sessile. Barnacles are sessile, especially as adults. What's it mean when I say an organism is sessile, by the way? What would be the opposite of something that's sessile, maybe? Another name for sessile might be something like this. I spell it with it. There we go. Anybody? Tatiana, what's sessile? What's sedentary? Olivia? Well, the opposite of sessile. So an organism that is not mobile is sessile or sedentary. Barnacles, oysters, clams are generally sedentary organisms, right? They settle down on a spot and they don't move. Very much different than four-legged creatures or the fish that are in this picture, right? They're mobile critters. Well, organisms that are sessile have very different reproductive challenges than organisms that are mobile. So we'll talk more about that later on, and I'll talk to you about how the barnacle deals with this, the barnacle being a sessile organism. All right, so back to allopatric speciation. Allopatric speciation basically can be summed up in one sentence. In allopatric speciation, what keeps the organisms apart, what keeps them from mixing, is a geographic barrier. Simple as that. By definition, allopatric speciation involves a geographic barrier. It can be something whereby in this picture the water level dropped and now the fish can't get over that middle hump that's in the middle. It could be something a little bigger, like when the Isthmus of Panama formed between these species of shrimp, right? And remember what we talked about in class? We said that at one time there were 30 species of shrimp, and when the Isthmus of Panama formed, we get these sister species. And I hope that you understand the significance of what I mean by sister species and how this, you know, how this is great evidence for allopatric speciation. What was it that divided Alpheus formius from Alpheus panamensis? And then the two green ones? See, what we're saying here is the ones in green are more closely related than the ones in red. Now, if you look at that for just a second, that seems a little counter to what you would expect, right? I mean, you would expect Alpheus formosus and Alpheus nuttingi, the ones up at the top, to be more closely related because they're on the same side of the isthmus. These shrimp don't make a long, there's no huge roving bands of shrimp moving across the Isthmus of Panama, you see. So, and the Isthmus of Panama forming in a relatively short period of time has allowed us to see a living laboratory. This is perfect allopatric speciation. The reason why Alpheus formosus and Alpheus panamensis are closer related to one another than the other two. So if I could kind of draw this on a family tree, I would say, all right, there's Alpheus formosa, or formosus. There's its sister species, right? And then the other two, the green ones, you see how I'm representing that? They're over here. Alpheus nuttingi or Alpheus milliceae. That's what I mean by sister species. So they're all closely related to one another, but these two are more closely related than these two are. And the only way we could demonstrate that, or the only way we could really explain that, would be that there's something that must have split this group of shrimp in half. Otherwise, how would Formosus be more closely related to Panamensis, which is on the other side of the Isthmus of Panama? Well, the Isthmus of Panama wasn't always there, right? So it's a geographic barrier. So a geographic barrier that can form, a geographic barrier, like, for example, in the river, that it cuts between the two species, or between that group of pine trees. The Grand Canyon, splitting up the chipmunks that I talked about. It can also involve species moving down around a geographic barrier as well. This would go back to the thing I said about mobile, right? Mobile organisms, as they disperse out in the environment, right? You know, the species kind of starts out here, and then as it disperses out, and the reasons, again, why organisms are dispersing is because it's all about resources. Parents want their offspring to get away from them because your offspring, I know we love our offspring, right? We love our kids. Little baby chipmunks are loved by their mama chipmunks, I suppose. I don't know. Anyway, but the point of fact is, even if we can say, well, we're not sure if chipmunks love their offspring, they have a genetic contribution in your offspring, right? That's a very biological thing. You want to take care of your offspring because they have some of your genetic information in you. You remember, I don't know, maybe you don't remember, but in Bio 1, one of the things you might have learned about DNA is that your DNA it cares nothing about you. Your DNA doesn't care about you. Your DNA only cares, if a molecule can care, about itself. It likes to make copies of itself. So what you do when you are taking care or caring about your offspring is you're actually preserving your genetic information, because your offspring are half of that, aren't they? As a matter of fact, my daughter and I were having an interesting discussion about altruism yesterday. You know, why would somebody, I could see you take a bullet for your kid, right? But why take a bullet for a stranger? And there are people who do that, because if we think about it biologically, you might take a bullet for your kid because you share some genetics, but you don't share genetics with a stranger. You don't share very close genetics. Now, family relationship. We could talk about this, but this is actually more of an anthropological thing. It has to do with us living in small villages at one time. But the point is that we do disperse out, and one of the reasons that we disperse out, of course, is to not compete with our parents for resources, or our parents not wanting us to compete for resources. So when the oak tree drops its seeds, it likes to have those little wings or some other device to carry those seeds. I meant maple, not oak, by the way. It likes to have these wings that carries those seeds off into the distance so they settle down someplace else. Because guess what? If they drop to the forest floor and start rooting and growing right below the adult tree, you're competing for resources. And while I didn't mind providing genetic information for this offspring, once that's done, especially in the case of trees, obviously, you're on your own at that point. So dispersal is important. It's also, of course, important to prevent inbreeding. I think I talked about that with the killer whales. All right, so up there at the top, though, a species can disperse, and as it's dispersing, can find a geographic barrier in the way that you have to go one side or the other side, so you're, again, limited by a geographic barrier. And now, depending on how long it takes for them to disperse around that barrier, how big the barrier is, I mean, they might, as they move their way around and combine back together on the other side, they may or may not have become two different species. In other words, they may be able to breed and produce viable fertile offspring just like there were before. So we would say that whatever the geographic barrier wasn't extensive enough. They weren't apart long enough. This is another sort of example of allopatric speciation, but this is sort of the opposite of what I said. It's not the creation of some geographic feature that separates the species, like a river or the Isthmus of Panama. It's not organisms dispersing out around a mountain. In this particular case, it's allopatric speciation because something in the environment physically gets removed. The trees are in the red X, right? Remember yesterday I was saying is these trees would all share genetic information with one another, even if the trees in the foreground and the background were too far away. Through a relay system, they would be reproducing with trees nearest to them, and of course that would connect to the trees in the back. So genetic information flows all the way through those trees, and let's say again something happened in those trees, a fire, a flood, a boulder, like with genetic drift, killed all these trees off. And if you kill all of them off, then what you essentially now have is two potentially separate populations. Okay? So you got to think about allopatric speciation, more just the idea of a river that cuts through a group of organisms and splits them into two sides. Or we can think about it also on islands, especially islands that have that are archipelago, or a collection of islands. All these little tiny islands out here, all of them represent possible places that founders can find themselves, right? We talked about the founder effect. So in this case, we're looking at kingfishers, and they're making their way from the mainland to some of the outlying islands. And on those outlying islands, because presumably, once again, which birds made it out there were just a few of them. Remember, once again, I got to make sure that you understand this. The founder effect is considered genetic drift. The organisms that make it out to those islands, these isolated island populations, again, these are the success stories. We defined the founder effect as genetic drift. In other words, genetic change, evolution that occurs by chance. We say that a founder effect is genetic drift because the original founders got to the island by luck, by chance. And the reason why I seem to harp on that is because students sometimes get that confused because they think, okay, founder effect, we're talking about kingfishers or Darwin's finches on the islands in the Galapagos. Those organisms, if they survive, they must have adapted to those islands. So the founder effect involves adaptation, natural selection, which it does not, remember. It's not what happened after the birds got there and how they changed. That is adaptation. Yes, that's natural selection. If they survive. If they survive. Remember, founder effect is chance. The birds that made it out to the island. You're standing there on the island, and you're there the day that Darwin's finch, or maybe a handful of finches arrived. Wrong word. They arrived. You're there, and here they come. Oh my God, where'd all these finches come from? I'm standing in the Galapagos Islands. We're hundreds of miles offshore. Maybe a storm blew them over. Here they are. We got 10 birds. We measure their genetic frequency. We measure their P's and Q's. We do a Hardy-Weinberg on them. We do the P-squared 2PQ just like we did in class. Now think about it for a second. If I'm standing out on that island, and I'm a biologist, and 10 Darwin's finches, just 10, could be just one like Myers said, but let's give 10 just for, whatever. 10 Darwin's finches, or 10 birds that became Darwin's finches, 10 finch ancestor birds make it out to the island. If I measured their genetic distribution, their genotype frequency, if I did the Hardy-Weinberg on them, would you think it would be the same as the birds on the mainland from which they came from, or would you bet that it would be different? So the founders, would they have the same genetic information in distribution, same number of heterozygotes, homozygotes, heterozygotes, recessive, just like we did with the Hardy-Weinberg? Do you think the values of P squared, Q squared, and 2PQ would be the same for those 10 that landed on the island while you're standing there versus all the ones they came from back on the mainland? What do you guys think? Yes or no? Yes, why with a question mark? Be firm. Yes. Now tell me why. Yes is correct, by the way, but now why? Why would they be different? No, it's not the equilibrium. You said they would be different, but if you said the equilibrium never changes, then they would stay the same, right? You're saying that the genetic frequencies, on the mainland, P squared equals this, 2PQ equals this, Q squared equals this. 10 finches, a couple hundred miles out to sea on one of the islands, coming from the mainland birds. If you calculated the P squareds, all those frequencies again, would you expect they would be the same or practically exactly the same as the one on the mainland, or would you expect they might be different? The other day I gave the example of we were trying to calculate average human height. and we did it with the 8 of us in the room, and we said, all right, let's take everybody's height, divide by 8, that's the average human height, and then tried to say, all right, on Earth, the average human is 5 something or other based on the data that we got in class with us 8 people. What would be wrong about that? What would be presumptive about us saying, oh, well, the average height of people in our class was 5'7", though we're humans. Throughout the world, the average human must be 5'7". What would be wrong with that? Again, those of you who've had stats class, think back to what I'm saying. I'm not doing a good job of explaining this, am I? How accurate? Yeah. There's a lack of accuracy if we only did it with 8 students, right? I mean, like I said the other day, maybe we just happen to get 8 relatively shorter than average students. And we might say, well, the average human height is 5'4", because in the class we had a lot of shorter people, or it could have been a lot of taller people. It's only 8 students out of 7 billion. So when those 10 finches arrived, holy cow, Steve, they arrived out to that island, and you were there and you calculated their frequencies, they represented the genetic information of 10 birds out of, oh, I don't know, say there was 150 million on the mainland. It's the same argument, isn't it? Those 10 birds probably don't represent the average genetic frequency, because 10 out of 100, it's like, again, we probably wouldn't do a very good job being able to calculate average human height by just measuring. I mean, if you want to make it ridiculous, we could say just 2 people, just me and another person. Go at home, I measure my height, I measure my wife's height, and I say, all right, divide by 2, that's the average human height. There's only 2 of us in the room. My wife and I, neither one of us are on the tall or the short end, so we might give a pretty close number. But my dad, on the other hand, he was over 6 foot. My mom was closer to about 5 foot, and the number would be a little different there. Cannot represent a population of people that all live in different areas. Yeah, exactly. Exactly. We're living in one part of the world. We've had different nutrition. There's different genetics. You can't say 8 students out of a Bio 2 lab at Lake Sumter, average height represents the average height of all the humans on the planet. It's too small of a sample size, right? That's the thing I'm getting at in statistics. So when you have founder effect, the reason why the initial frequencies, when those founders get to the island, the reason why those have changed through drift is because they probably don't represent the big population back on the mainland. Now, again, here's where students make the mistake. They want to immediately equate founder effect with adaptation because the founders survived. Yes, if they survive, then it becomes natural selection. Then it becomes adaptation. I'm talking about the initial frequency of genotypes when it got there. That's chance, right? That's the founder effect. Okay, questions on that? All right, so that's all going to fit with habitat isolation, because that's all that really is. The founder effect says that the habitat is that the expanse of water between the mainland and the island is big enough that the birds don't typically make it there. They can, but don't typically. In other words, it's a rare luck event, if you will. Reproductive isolation in this case, whereby the environment doesn't necessarily create a barrier, but it removes a link, right, with those are there, or as we talked about, obviously the river as well as the organisms moving around a mountain. Those are all habitat related. So if someone were to say, well, what type of reproductively isolating mechanism is involved in allopatric, there's only one: habitat isolation. That's it. There's only one. Here's where it gets a little bit trickier, because in the next type, which is not allopatric but is sympatric. See the one on the right? Sympatric speciation involves the fact that that an original one species of these fish, the ones in orange, through time we get more than one species. We get the orange ones, right? And then we get the blue ones. Now, it might be that the orange ones evolved and become another species of fish, but at the same time somebody else split off and became the blue species of fish. So we might have a situation where it's like that, right? So these ancestors, these would be the ancestors, gave rise to 2 new species. Or we could have that the ancestral population kept going along. So these are the orange ones. And somewhere along the way, the blue ones branched off. See what I mean by that? So we're not necessarily saying that from the picture up above to the St. Patrick one, we got, 2 new species out of this. We might have got 1 species that was already there and a new species on top of that. All right, makes sense? Obviously, this is not going to involve habitat, though, is it? Because sympatric speciation, allopatric speciation means speciation in another place. That's what allo means. Allopatric is another place. Sympatric speciation, like sympathy, or even the word synapsis. Well, there's a good blast from the past. What is synapsis in biology? What is synapsis? Sometimes it helps to learn the roots of words. So when you see words that maybe first time you might see them, you can maybe figure out what they're talking about or help to remember. Does anybody remember synapsis? I hope so. It happens during prophase 1. Synapsis. Yes, okay, when they lay next to one another during crossing over, and they exchange genetic information. We call it synapsis because the word synapsis means to lay side by side. You get the 2 sets of chromosomes, the homologous chromosomes. I'm sorry, maybe that's a little bit better. Laying side by side, they are in synapsis. SYM means next to. So if you have sympathy, you have feelings that are similar to someone else. Gosh, it's really bad. I heard about your mother dying. My mother died. I got sympathy. I know what it feels like. Sympatric speciation then involves species that occurs right next to the original species. You're not in someplace else. You're not geographically isolated. So this is going to involve a lot more of the behavioral and the temporal and a lot of those other things. It's not going to be a physical barrier. It's going to be something, very oftentimes it's going to be something physiological, or it's going to be something behavioral. Let's look at it in plants first. Sympatric speciation is well known in plants. It's also known in animals, but it seems to be more so in plants. So let's take 2 species of plants with different chromosome numbers. Species A has a diploid number of 6, and species B has a diploid number of 4. So here's what we're trying to say. This is a possible way by which we could get a completely different species. Now, I'd like you to jump ahead of the picture for a second down to the very bottom. This is what we're going to make at the very bottom. We're going to make a new viable, fertile hybrid, all right, with a different chromosome number of 10. So how do we get that? Okay, well, let's take a look. Species A undergoes meiosis. Species B undergoes meiosis. Species A undergoes meiosis. Everything's fine. Three chromosomes in one gamete for species A, 2 chromosomes in the other one. We went from diploid to haploid. So far, nothing new, right? Everything we thought about. Now, although gametic isolation is something that's very important in terms of keeping the wrong sperm from the wrong egg, Mother Nature's not always perfect, right? Mother Nature doesn't always get it right every time. And among plants, and it does happen in animals as well, but among plants, there is a situation where occasionally species A and species B, the gametes of those 2 different species will fuse. Now, let me qualify that just a little bit. Species A and species B are relatively closely related. They may be 2 members of the same genus, like Canis familiaris, the dog, and Canis lupus, the wolf, right? So I'm not talking about, in case of plants, a pine tree and a moss. We'll learn a little bit later on that those organisms are very different from one another. So we're talking about maybe a couple species of grass, right? Maybe a couple species of grass. All right, so we get a hybrid that is sterile because it's got 5 chromosomes. The sterility involved in that is because you got an odd number of chromosomes, right? You got to have even numbers of chromosomes in order to have a fully functioning human being, now, or a fully functioning organism. Now, in the case of plants, this sterile hybrid survives, okay? Again, it's the exception, right? Many times this is not going to happen. These gametes will never fuse, and even if they did, the hybrid wouldn't survive. But let's say in those rare exception cases, which it does happen sometimes, that sterile hybrid does survive. Now, that sterile hybrid undergoes a mitotic or a meiotic error. What we mean by a mitotic or a meiotic error, especially focusing in the meiosis side of it, is what happens to the number of chromosomes. I'm not talking about errors that mess up the chromosomes or get the wrong chromosomes to line up with each other. I'm not talking about any of that. I'm talking about a situation where when chromosomes are supposed to separate, you remember during anaphase and mitosis? You remember during anaphase 1 and 2 in meiosis? That's what's happening. Chromosomes are splitting apart. But let's say some error occurs where they don't do that. Now, in Bio-1, you should have learned about how this comes about. If you'll stick with me a second with my limited drawing abilities. Let's take a cell and just a pair of chromosomes, and they're supposed to split and go to either side. Does anybody recall the name of the structure, the cellular structure that was responsible for pulling those chromosomes apart? Anybody remember, like where I drew the arrows? What is the name of that structure that indicates what I'm drawing with the arrows? What pulls those chromosomes apart? Let's see how you remember your Bio-1. Nobody, huh? Suzanne, how about you? Sharon? Nope. Olivia? Nerali, what are those little things called that pull them apart? Yeah, that's the stuff at the end of the chromosomes. That's really telomeres are cool. Telomeres at the end of your chromosomes that prevent your chromosomes. Well, they don't prevent anything. They get shorter and shorter as you get older. No, remember spindle fibers. Those are spindle fibers that pull them apart. Now, and this does happen. What happens if a mistake occurs and you've got these two chromosomes, but a spindle fiber forms on one, but there isn't one on the other side? You remember that? That would have been in the discussion toward the end of the semester when you were talking about things like trisomy 21. Right? So you get an extra chromosome in that cell because if that spindle fiber doesn't form. The name of this, if you remember, was chromosomal, I love this word. It's one of my favorite words in science. Chromosomal non-disjunction. In other words, the chromosomes don't disjunct. They don't pull apart when they're supposed to. So instead of each cell getting a copy of the chromosome, one cell gets two, right, and the other cell does not. Remember, back in trisomy 21, again, in Down syndrome individuals have 3 number 21 chromosomes. That's how this happens. The egg or the sperm, during meiosis, instead of the 21st chromosome separating, going from diploid to haploid, those chromosomes don't separate, and you end up with a gamete that has 2 21 chromosomes, and then the other gamete brings in its one, and you got yourself 3 21 chromosomes. All right, so that's what I mean by a mitotic or meiotic error. And in this particular case, what happens is the chromosomes replicate, but they don't pull apart. So now you have a new species, a new viable fertile species, a new viable fertile hybrid with the chromosome number of 10. Completely different species. Okay? Now this is what we call allopolyploidy. Allopolyploidy, define them there on the right for you. Allopolyploidy is when you get a speciation event that occurs between 2 separate species. All right? Species A, species B. Allopolyploidy. There's another type called autopolyploidy, and in autopolyploidy, what happens is it occurs within a species. So species A actually goes through a meiotic error where it produces a completely different set of chromosomes. So it's not 2 separate species, it's species A through these chromosomal mutations, or not mutations, excuse me, through these meiotic errors or mitotic errors causes an organism with a different number of chromosomes. It's what we call polyploidism. You might have heard that in Bio-1. Now that's autopolyploidy, all right? Now, plants do this rather regularly. Well, not regularly. They do it more regularly than animals. And more importantly than that, it appears among plants that changing chromosome numbers, either through autopolyploidy, within a species or between species, and allopolyploidy, there is, how shall I say this? It's not as immediately a death sentence as it is in animals. There's not a lot of animals that can tolerate polyploidism. There's not a lot of animals out there that can tolerate allopolyploidy or autopolyploidy. What I mean by that is typically in animals, when gametes are malfunction like that, they produce ill-formed embryos that are never carried to term. They never hatch. They never survive. Or if they do survive, they're not viable. Plants seem to be able to get away with this a little bit more. And that's actually good for us, because we have found out that in agriculture, we can stimulate plants, closely related species, to engage in these polyploidy events, to actually add the number of chromosomes to take wild species of plants and turn them into things that we can eat. For example, bread wheat on the right-hand side, the stuff we make bread out of, is related to a wild grass. There it is on the left-hand side. That's where it came from. So on the left is the natural wild grass, and on the right is the bread wheat that humans throughout history, and in more recent history, when we understood all this, we used selective breeding techniques and this whole idea of allopolyploidy, because, and I don't remember what the chromosome number is exactly, but bread wheat has got more chromosomes than the wild grass that it descended from, or that it evolved from. And again, that's because through allopolyploidy, that bread wheat has added more chromosomes. Again, does not happen in animals as often. It's usually detrimental in animals. So animals, when they undergo sympatric speciation, polyploidism is not the typical way that happens. All right, they're not typically, animals are not going to create new species by changing their chromosome numbers. Remember, I talked about that with you guys. Changing chromosome numbers or changing structures on chromosomes generally is death. Generally is detrimental to the critter. But let's take a look at way at some of the animals can engage in sympatric speciation without messing with their chromosome numbers. Well, again, it's a behavioral thing. So let me tell you the results of a study in which we had got a very interesting result having to do with behavior. In Africa, Lake Victoria, there are some fish in Lake Victoria called Lake Victoria cichlids. In this particular case, in the slide you see here, these are all members of a genus known as Pundamilia. There's Pundamilia pundamilia, and there's Pundamilia nairi. So on the left-hand side is the species I'm mentioning. And if you look at them under normal light, they look different. Now, body shape, they don't look very much different. But then a lot of you might say, well, gosh, not a lot. Fish kind of look like fish. All right, I'll give you that. But when it comes to body coloration, there's quite a clear difference, isn't there? Right? Pundamilia doesn't have no orange, really, a little few blotches of yellow. Whereas nairi, the stripes are darker, the overall body coloration is darker, and there's definitely a lot more orange there. So when in the wild, when out in Lake Victoria, Pundamilia pundamilia and Pundamilia nairi, these are just the males. The females who do the selecting, like we talked about, female Pundamilia, let me call them species 1 and 2 so I don't keep tripping over my tongue. Species 1, species 2. Females of species 1 will pick males of species 1, and females of species 2 will pick males of species 2 based on looks, based on simple morphology, right? So they're doing the same thing we did when we said, oh, horse and dog, right? They're doing the same thing. So females of each species will pick males that look like them. So biologists, ichthyologists looking at this wondered, is there really something significant about the difference between these two males of these species? In other words, why don't we get hybridization? What is the reason that these organisms couldn't breed with each other? Because they are closely related to one another, both in the same genus. So some ingenious scientists had this great idea. They said, what if we take them into a laboratory and we throw them into an aquarium, and in the room where that aquarium is, we only illuminate that room with one colored light, an orange colored light, a monochromatic orange colored light. Now, if you look on the right, this is what males look like under the orange light. throw the females back into the mix. Are the females only selecting the males on the way they look? Or maybe we could reword it and say the hypothesis. Oh, is that right? Yeah, because they get good size. You can eat them. Oh, that's wonderful. I went to Kenya. I went to Mombasa many years ago. I remember the thing I remember most distinctive. Well, I remember many things about Kenya and Mombasa. Don't get me wrong, but I was in the Navy, and as my ship was pulling in, I was out on the lookout deck, and I had my binoculars, and I picked it up, and there was a bunch of folks gathered on the dock as the ship pulled in, and the fella had a Domino's Pizza shirt on, and I thought, my goodness, Domino's Pizzas all the way over here in Mombasa, Kenya. There was no Domino's in town, so to be honest, I'm not sure where he got the shirt, but probably some tourist or something like that. But yeah, I love Africa. Incredible. All right, so but back to these fish, these females. They put them into the monochromatic orange-colored light. Females bred with males of either one produced viable, fertile offspring. So now my question to you is, based on what I just said, are they two species or one? Doesn't matter if a female, yeah. But how come they have two separate names? Two separate names tells me they're the same species. But you just told me they're one. So, not a male versus female thing. It's no different than the dogs and the wolves. Dogs and wolves have different scientific names, don't they? Horses and donkeys have different scientific names, don't they? But even though dogs and wolves, especially, have two different scientific names and ostensibly are two different species, which is the whole reason behind the scientific name. Well, I'm just saying, Tatiana, the reason why the females could breed with either male and produce viable, fertile offspring is just like Nirali was saying, they're really one species. We have them two separate names, but remember, animals, organisms don't come into the world with their scientific name tattooed on them. We, humans, give them the scientific name. So, whereas Aristotle would say Pundamilia is on this part of the ladder of the ladder, and Nairi is on above or below, doesn't matter, is on another rung of the ladder. We can show quite clearly that it's very possible to be in between the ladder. That the only reason that females are not selecting males of the other species is not because of anything where they can't produce offspring. It's no reproductively isolating mechanism physiologically. That's it. They're just separated by looks. It's a behavioral thing. Young female Nairi females growing up being taught by their Nairi parents that these are the males of your species, and the same with the Pundamilia. But when it comes right down to it biologically, there's no difference between these. They reproduce and produce viable, fertile offspring every time. Not just in the laboratory either, by the way. Here's one from the same lake. This is a Pundamilia from the same lake, caught in the wild. That is a hybrid. You can see it's actually got a mixture of both the white and the orange, doesn't it? Now, interestingly enough, these hybrids only occur in areas of the lake where the water is turbid. You see the name right there, or see the word, turbid. What does it mean when water is turbid? That's exactly right. Now, elaborate on the environment. What about the environment? Okay, what about the environment causes these organisms to either pick the male of the same species, or in this case, obviously, a male and a female, two separate species, out in the wild? That's right, water is cloudy. It's visibility, isn't it? So this is a pretty feeble isolating mechanism. When we talked about habitat isolation as being feasible, or as being feeble, in other words, easy to overcome, this is another one. The only difference these two organisms have is the fact that the females are picking males that look like them. Otherwise, they could breed with males of the other ones and produce perfectly fine offspring. Now, 100 years from now, 1,000 years from now, 10,000 years from now, maybe never, it all depends, would we eventually have two organisms that are so far apart that they couldn't produce viable fertile offspring? Yeah, I mean, here, if we're going back to my little thing where I use the branch and the tree, even though they both have different scientific names, you would think they're on different branches of the tree. Technically, they're both right there. We just give them two separate names because out there in the wild, the females tend to mate with only those. But given an opportunity to be tricked, if you will, or to not be able to tell the difference between the two, they mate with either one. So this is sympatric speciation, is it not? Right? We ended up with these two species out here while they all lived in the same environment. Now, the only different in the environment would be the turbid versus the clear water, but that's not the same kind of geographically isolating barriers we saw in allopatric, okay? So we've been able to document sympatric speciation in fish. We've been able to document in amphibians too. Both of these organisms here occur in Florida, Hyla chrysochelys and versicolor. And they're two separate species with all of the designations of separate species and all that stuff that we know about with species. But the interesting thing about these two critters is the number of chromosomes in Hyla versicolor is exactly the double of the amount in Hyla chrysochelys. So if we go back and we come up with a reasonable hypothesis, we suggest that what happened is among some Hyla chrysochelys, a male and a female, some meiotic error occurred, and instead of producing haploid gametes, they each produces diploid gametes. Now, this is not something that's totally unheard of. In Bio 1, you probably learned about triploid and tetraploid. Triploid individuals having 3 N chromosomes, like in human beings, that would mean 69, and that does happen. Those folks obviously have physiological and other types of severe problems. In some cases, you can even be tetraploid and have 92 chromosomes. Again, those folks generally, or not generally, but those folks are going to have all sorts of physical and medical issues. Among amphibians, not quite as much, though. In certain lines of amphibians and in certain cases, it can be tolerated. So the research seems to indicate when we look at the genetic information, beyond just counting the numbers of chromosomes, that that's where Hyla versicolor may have come out of. It was a sympatric species event by which a mistake in a male and a female Hyla chrysochelys, or maybe in a bunch of them, created diploid sperm and egg, and there's Hyla versicolor. Now, within Hyla versicolor, the chromosomes get divided properly, but the change between the two species. All right, so this leads us to this whole idea of hybrids, right? We talked about hybrids. Now, these are not hybrids. You got one species from another. That's a hybrid. We've taken a look at that, and we've talked about hybrids. We've talked about hybrids being sterile and hybrids not being sterile. Now, as I said to you yesterday, biologists love hybrids, man. Especially biologists who study population evolution. The reason being is that once I said before, hybrid zones and areas where hybridization is occurring in the wild is a living laboratory. So look, let's kind of take this general picture first, and we'll look at a couple of examples. So we got on the left-hand side, we got a population, all right? And what we're looking at here is those 3 little dots are going to represent 3 populations, but they're all close enough to one another, the genes flow between them. So they basically may be 3 separate populations physically. One lives down by the river. One lives out of, maybe up higher, and one lives maybe more on the flat plain of the floodplain of the river, whatever the case may be. They're close enough to one another that genetically they're one population, though. So if you did the Hardy-Weinberg on them, the calculated values would be for all 3 of those to get together because, as I said, the genes flow between them. And then we move along and we get a barrier to gene flow. All right, now don't get fooled or confused by what we mean by a barrier to gene flow. We get one of those isolating mechanisms. Gene flow, again, is mixing up genetic information typically through reproduction. If you're not mixing up genetic information, there's no gene flow. So that gene flow, of course, we think about gene flow as organisms moving in and moving out. That's how they get there, or that's how they leave. The end result is when they move in, they bring in their genetic information when they breed. When they leave, they take their genetic information away from them. or with them. So again, we get a divergence here. We have one of these population that for whatever reason could be allopatric, could be a physical barrier, could be any of the things we've talked about, could be a sympatric speciation event, a behavioral change, whatever the case may be. We get a population that diverges, and it diverges, and it diverges, and it gets different, and it gets different. And physically in between, there's a hybrid zone. So when we think about hybrid zones, when we have two closely related species, and they meet physically in this hybrid zone, the part that's in the purple there are three things that can happen. Number one, you can get reinforcement. All right, now what happens in reinforcement is we reinforce the species being different, and the hybrids, the part that's in the purple arrow there, this represents the hybrids. The hybrids, although they might be viable, although they might be fertile, and although they might be hybrid and fertile, they may not be as fertile, and they may not be as viable. So the reason why the purple arrow is getting smaller going to the right, as we move to the right, we're indicating that while hybrids happen, there's not a lot of them. And it might even be to the point where they start to peter out after a while. You start losing the hybrids. All right, so you get reinforcement. We're reinforcing those two populations staying away from each other. Now, what will eventually happen if the reinforcement continues? These might eventually diverge into two different species, or one if one of them was the original species. Another thing that might happen is these areas of hybrid zones, these organisms that are found in the hybrid zone reproduce and produce viable fertile offspring, and then after some period of time, the populations fuse back together, whatever this barrier to gene flow is removed. This would be often in cases like something physical, right? A river that cuts between two populations, they're apart from each other. The river lasts for, it's a small creek, 50 years, and then whatever happened, it dries up. All right, so there's not a river there anymore. So now there's nothing that blocks the organisms moving back and forth. They've only been apart 50 years. They come back together now and they fuse. So hybrids are short-lived. There were hybrids for a while, but now whatever keeps the gene flow apart is no longer there, so they fuse back together. If hybrids stick around, if what you get in the population are species A, species B, and a bunch of hybrids in between, then it's stability. So in this case, the purple thicker arrow represents the fact that hybrids keep moving on into the future. So what kind of world is that? It's a world with species A, species B, and the hybrids. It's a world where you have, do I need to go back? These things. See, this is a hybrid of those two fish that's found in turbid water. Well, as long as there's going to be turbid water in that area, right, you're going to have males and females, or females more specifically, that make mistakes and mate with the other male. So the hybrids are going to be maintained. So that's going to be an example of stabilization, or we're stabilizing the hybrids. So we get two species, they stay two species, yet the hybrids still remain around. All right, so reinforcement, fusion, and stability are the three possible outcomes when you look at a hybrid zone. Reinforcement, the hybrids die off and diminish. Fusion, hybrids are temporary. Stability, hybrids are permanent, more or less. All right, let me show you an example from the wild of what I mean by this whole idea of reinforcement, of actually reinforcement, an example of reinforcement in a hybrid zone. All right, these are what? Flycatchers, that's right. In Europe, we have two, in this area, two flycatchers, two species of flycatchers we're mostly interested in. In the green area, we have the pied flycatcher, and these are all males of the pied flycatchers, all right, and what the males look like. And down near the southern part in the blue, we have what's called the collared flycatcher. And in between, there is a hybrid zone in yellow. So pied flycatchers and collared flycatchers mix in that hybrid zone in between. All right, now, how do the females keep it straight? Because here's the thing, females are doing the selecting on these birds. When a female is in a hybrid zone, all right, when a female is, and I'm going to make sure I get my notes right on this because this is something that I always have to be careful I don't say backwards because it can be confusing. So when females are in the hybrid zone, okay, so when females are in that yellow part, they will occasionally make mistakes. All right. So if they have a situation where a female pied flycatcher is flying in the yellow, especially the more towards the middle of the area there, she might have a mistake of mating not with another pied flycatcher, because look how the males look in the hybrid zone. They look a little brown, but she might make the mistake and breed with a collared flycatcher. Collared flycatcher looks a little more like her, especially during feather colors. Now, it's the same for the collared flycatcher when females are in that yellow region. They'll mate with, occasionally make a mistake and mate with a pied flycatcher. So that's where you get the hybrids from. What's interesting about that is those mistakes only happen when females are in the midst of that hybrid zone. Female pied flycatchers up in the edges of where the hybrid zones are, and female collared flycatchers rarely make the mistake. They usually always mate with the right male. It's in the middle where the mistakes happen. All right, so this becomes a behavioral issue. Since the females are doing the selecting, right, they're behaviorally selecting based on appearance. But for whatever reason, and you'd have to look into the visual acuity and color, the ability to tell color and all this sort of stuff amongst these female birds. But when they're in an area where there are the pied flycatchers and the collared flycatchers, and the pied flycatchers look brownish and the collared flycatchers look more like what the female pied flycatchers are used to, they tend to make mistakes. Now, the reason why this is considered a reinforcement of a hybrid zone, in other words, in reinforcement, pied flycatchers stay one species, collared flycatchers stay another species. Even the ones in the hybrid zone with different colors are still that species. The reason why this is reinforcement, though, is because hybrids in the area of yellow, while they do exist, are rare. They're not quite as fit as the parents, we believe. The females are doing the mate selection, and what we see in this example is females doing a mate selection in the yellow area have a better chance of making a mistake. So the hybrids that are being created in the yellow area are hybrids that come about by mistaken identity. Now, if the hybrids that were produced were strong and viable and all that sort of stuff, then we wouldn't have reinforcement. We would have stability, right? We would have the bottom result, right? We would have stability. Hybrids are maintained, and so are the two species. We're saying here that the hybrids are diminishing. Hybrids are diminished. Hybrids are rare. They're not as fit as the parents. They're not going to ever raise to the population of the parents. Okay? This is reinforcing to keep the two species separate based on behavioral aspects. The only time when females make mistakes is only when they're confused in the middle zones. All right? So if for some reason a male collared flycatcher flew way up in here into Scandinavia, a female pied flycatcher would not mate with him because she's got the normal looking males around, the pied flycatchers. It's only in the yellow zone where those females make the mistake. The next example having to do with this has to do with allopatric speciation that I talked about when it comes to a physical geographical barrier. The North American salamander. This is an example of what we call a ring species. Ring species are called that because their distribution surrounds something like a ring. All right, so follow what's going on here. Moving up to the top, we have the original population of this salamander in Santia and Schultzi, right? That's the original Oregon population. Of course, salamanders like everything dispersed, and as they dispersed out of Oregon into northern California, they came across the mountain range in the middle of California known as the Sierra Madre Mountains. So salamanders, not being great mountain climbers, for the most part, what they did is they went around either side of the mountains. And so we ended up having two groups of these salamanders. We had the yellow-eyed and the Monterey, which were the coastal populations. And then with the Sierra Nevada, the yellow blotch, and a little later on, you'll see the large blotch salamander. Those are in the inland populations. Now, in the little green boxes there. So the populations diverge. Look what's happening where I point to number 2. In that area in the middle in the green, whenever the inland and the coastal populations do meet with one another, let's say in valley areas or just areas where just for whatever reason those salamanders do actually come close enough they can mate, they do occasionally breed with one another and they do occasionally produce fertile hybrids. So fertile, viable hybrids. So if you think about the salamanders moving down around the mountains as being like in time, it takes time for them to do that. You can see, or you can agree that it's not too surprising that in this area here, they're still not separate enough from one another to make completely different offspring, right? In that area there, they've only been separated from each other as long as it took for salamanders to move down around that part of the Sierra Nevada mountains. So if they do come across each other, they can still produce hybrids that are fertile and viable. But when you get a little bit further south and the ring kind of comes back together, and you get things like, for example, large blotched inland salamanders, these guys, found in the same area with the Monterey or the other coastal populations, you get no hybridization. So once we've moved a little further south and you start to look at those populations like the yellow blotched and the large blotched and the coastal populations, now they've been further away from each other long enough that now they do not hybridize. So this is kind of seeing speciation and hybridization in a time scale based on distance more than anything, not just time, but what we have is organisms moving around a geographic barrier. So this would be a good example of a ring species caused by allopatric speciation, right? The mountains are providing the allopatric speciation. Great living laboratories. That's what we know happened. Looking at the genetic analysis, we can document that coastal populations and inland populations up around here in the green are hybridized and not that much different from one another that they can't, but once they have continued to move further south. Now, of course, the idea is salamanders can more easily breed with each other across this distance than the Oregon population could breed with the yellow blotched, right? So as they move down around the islands, the differences from each other increases. All right, any questions on that? Oh, I got just enough time to finish my last part. I'm going to jump 2 slides and then go back 1 slide because I want to do it in a different order. Fossils aren't common. We've talked about that. We talked about that in class, right? Spent all my life looking for fossil, never have seen one. So one of the problems and one of the things that Huxley talked about in that quote at the very beginning of this chapter, there's widely distinct gaps. Remember, Huxley said that if it could be proven that the differences between groups of organisms always existed, if I went back in the fossil record with Lamarck and I went, oh, here's 2 species, go back a little bit further in time in the rocks, there's still 2, never did they come from a common ancestor. That's fatal to Darwin's evolution. They're completely fatal. So one of the things that would puzzle paleontologists, especially early on, not so much in terms of the theory, but where they were, was where are transitional creatures? So, for example, in a very large scale, we know that a type of dinosaur, a specific type of dinosaur, through evolutionary change gave rise to modern birds. Now, remember I said in the first day of class, birds are reptiles. We're going to consider them reptiles, but what I'm talking about here is changing one type of reptile, a very specialized type of reptile, a dinosaur, to a completely different type of reptile, a bird. I could make the same example if when we actually get into subsequent chapters, when we talk about amphibians and fish. Amphibians evolved from fish. Reptiles evolved from amphibians. Mammals evolved from reptiles. So in all of those, there's going to be some part mammal, part reptile, part amphibian, part fish. And we'll talk more about that later on when we get to those groups. I'll show you those transitional forms, but that's what they're called. Where are these transitional forms? All right, so if we take a dinosaur on the left, and I just picked the dinosaur at random. I don't know if this was the dinosaur that was in the lineage that gave rise to birds, and then your common bird on the right. We would expect that there would be critters that were somewhere in between a reptilian or a dinosaur reptile and a bird reptile. All right? We would expect them to be there. And for many years, looking through the fossil record, we found plenty of dinosaurs. We found plenty of birds that lived around during the dinosaurs in the fossil record, but we didn't find something that really looked part bird reptilian or dinosaur reptilian. Okay, or if I go back to my thing with we didn't really find anything that looked like it was, say, for lack of a better way of describing it, half amphibian and half reptile, or half fish and half amphibian. We had no evidence of what we call transitional fossils. All right, well, we do now. And there's a reason why, and I'll explain it to you. It has to do with the punctuated equilibrium. But let me show you with this example. That critter in the middle that you see there, that is a fossilized critter. These organisms, this Archaeopteryx, has been extinct for millions of years. Down below is kind of what Archaeopteryx looked like. Now, what did Archaeopteryx look like? Archaeopteryx looked like a lizard with feathers. All right, it had a lot of reptilian characteristics. Claws like reptiles. It had teeth like reptiles. Birds don't have teeth. It's a big thing about birds. Ducks have teeth-like structures, but they're not really teeth. Birds don't have teeth. Reptiles definitely have teeth. A lot of the other anatomical structures, a lot of the skeletal structures were intermediate. In other words, the skeletal structures in the Archaeopteryx fossil, in some cases, looked like a skeleton from a reptile, look like a skeleton or from a dinosaur reptile, look like a skeleton from a bird reptile. So, like I said, this is one of these transitional forms. Now, when we talk about birds later on, you're going to find out that transitional forms, just so we don't give too much to Aristotle here, it wasn't like dinosaur reptile, Archaeopteryx, bird reptile. There were lots of transitional between the dinosaur reptiles and the bird reptiles. Archaeopteryx is just one of the first ones that was found. And Archaeopteryx fossils, like you see in the picture up above, are very rare. There's only, I don't know how many, but just a few handful of Archaeopteryx fossils on Earth. I was so excited when I went to the American Museum of Natural History years ago. They had an Archaeopteryx fossil. I was fascinated to be able to see it up close. But based on reconstruction of that fossil, we can say this was a reptile that had very reptilian, dinosaur reptilian characteristics as well as bird reptilian characteristics. Now, it probably didn't fly like a regular bird. As a matter of fact, we think it may have been able to fly more like a chicken, right? Feathers, and we'll talk about this here not too long, feathers didn't evolve for flight. Feathers evolved for another purpose first. They actually evolved for warmth. So the first feathers that evolved in organisms, these reptilian birds, were actually evolved not for flying, but for staying warm. So they didn't do a very good job of flying, in other words. All right, so Archaeopteryx, so transitional fossil forms. Why was it so difficult to find these transitional fossil forms? I mean, we found a lot more now. We found a lot more in all those other groups I mentioned too, which I will bring up when we get to those groups. But transitional fossils, although still quite rare, we have found quite a bit of them now. Well, how can we explain why there's so few transitional fossil forms? Well, it has to do with this thing called punctuated equilibrium. So I'm going to get myself a blank board here so I can draw this out for you. Here's how punctuated equilibrium works. Let's use a graph. Graphs are always nice. Let's say time. Okay, and over here, I don't know, some measurement of change. Genetic, morphological. One species and evolving, and we end up with another species. All right. This is what Darwin didn't understand. This is why the Cambrian explosion bothered him, by the way. See, Darwin's idea of evolution is the fact that evolution works in a very gradual pace, that changes that accumulate very gradually. Okay? Unfortunately, I can't erase just that line, so you're going to have to leave it on there. But here's what punctuated equilibrium says. Punctuated equilibrium says that evolution has two paces. Punctuated equilibrium, the name is perfect. Here's what it says. There are times in a lineage of an organism with little or no change occurs. You see, I'm going along time, but I'm not moving along the y-axis, so we're saying there's no change in that lineage. And then in a relatively short period of time, geologically speaking, there's a lot of change. And then that settles down back into an equilibrium where we go through periods of little or no change. And then that might be, again, punctuated with periods of rapid change. And then maybe that settles back down into periods of little or no change. Now, I want you to understand clearly. I don't want you to make the mistake. The line that I drew, this one here, gradualism, as Darwin understood evolution, does happen. But what Darwin didn't realize, and would have helped him with the Cambrian explosion, is sometimes speciation events occur pretty quickly, relatively speaking, folks. So if the species didn't change for 100 million years and then it changed quickly in 2 million years, right? We're still talking millions of years, still plenty of time for change to happen. But in the overall scheme of the history on Earth, organisms that were transitional lived and died and made fossils for about 2 million years. The organisms that came before, and I don't know, let's say the organisms that came after, lived for hundreds of millions of years longer. So it's a numbers thing, isn't it? Transitional fossil forms weren't non-existent. They were there. There's just not very many of them. Because these critters, these Archaeopteryx critter, these half amphibian, half reptile critters that I've been talking to you guys about, the half's not the right word, but part reptile, part mammal critters that I'm talking about, didn't stick around very long, relatively speaking. So they didn't have as many. There weren't as many to die, weren't as many fossils to be created. So when you're out looking around for fossils, which we've talked about the fact that they aren't very common, you got a much better chance of finding a fossil, one of these guys down here that was around 100 million years, or maybe one of these guys that was around 200, than the relatively small handful of those that was around only 2 million years ago. So this is what we mean by the pace of evolution. It can be gradual, right? And or it can be mixed in with periods of rapid speciation, punctuated equilibrium. Now, this is a relatively new, Titania, I'm sorry, did you raise your hand? I'm sorry, I was looking at the other screen. Punctuated equilibrium is a theory that was only put forth about 1972. Yeah, those are, yeah, there you go. That's right. At that time, when you see these rapid changes, the environment has changed relatively rapid. So environment is relatively, it changes a little bit there for 100 million years and then changes very quickly over 2 million years. You know, like the Isthmus of Panama formed 9 to 13 million years ago, but it only took a couple million years to form. Right? You got to think about it in relative changes. So yes, you're right. Environmental changes would cause these punctuated events to occur, but it would environmental changes in which the Earth changed, it at least for that species in that area, relatively rapid. I'm sorry, if I'm on the other screen, I can't see that you raised your hand. So and you can always write on the screen as well. Does that make sense? Could involve all of that. Yeah, competition is part of the environment. Remember, the environment is the resources, the weather, who you eat, who eats you. Yeah, your environment is everything you interact with. Everything you interact with. Make sense? Anybody got any questions on that for me? Neat. All right, one more slide so I can show you what this looks like. Got to go all the way back to those. Hold on. Clicking faster than the computer can keep up. Let me get that last slide to show you, because I want to show you what this looks like when we compare gradualism to punctuated equilibrium on something we call a cladogram. Sorry, my computer's not really. Let me go back. All right, so this is the slide that was just before this one. There we go. All right, so here are the two paces of evolution. It's not a question of is it one or the other. It's both of them at different times. So there's gradualism. So we start at the bottom of the bird, and through time there are gradual changes. Some reproductively isolating mechanism, could be allopatric, could be sympatric, whatever the case may be, but we get a gradual change in two lineage. So at the end of time, or at the end of the time of examination, we now have two separate species. All right, that does happen. In punctuated equilibrium, what happens is we have an organism, and maybe a species that never changes, right? But at certain periods of time, these branches that you see coming off there, we get a rapid speciation event. Now, this picture is not quite as extensive as it could be, because what happens sometimes is we don't just get one that comes off of there. We might get several species arising off of there at one time. So this little part right here would be the punctuated part. So periods of rapid speciation, see on the time axis, the orange and the yellow bird look like they're about the same time. They couldn't have been, but they might have arose just within a few 100,000 or millions of years. And then it happens again, and then happens again. But you see at the end, what happens with this branching is with punctuated equilibriums, we get a lot of diversity a lot faster. And I know, oh shoot, I'm going over time. I'm sorry, so I'll stop. But this is what caused Darwin problems with the Cambrian explosion. The Cambrian explosion in the fossil record, all of a sudden there was all these species. He only stood gradual change. He didn't understand that speciation can also be very quickly through punctuated equilibrium. All right. Punctuated equilibrium is also known as, and then I will let you go. I have to give you this one word. Go back to my chat. Punctuated equilibrium is also known as cladogenesis. So on the right-hand side is cladogenesis, to make different groups, and gradualism is sometimes also known as anagenesis. Oops, genesis. Cladogenesis on the right, anagenesis on the left. Again, not one over the other, they both occur. It's a speed. All right? The pace of evolution with punctuated equilibrium explains the absence of transitional fossil forms because, again, if these speciation events happen very quickly, the critters that are involved in them, the transitional ones, won't be around very long. All right, that's all I have for today. I'll go ahead and stop the recording. If you have any questions, by all means. If not, y'all have a good weekend, and I will see you in class. I will see you in class Monday. So get started on your assignments, your field notebooks, and let me know if you're having any troubles.